6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and the xylem. The xylem and phloem together constitute vascular bundles. Figure 6.5. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundles, because of the presence of cambium, possess the ability to form secondary xylem and phloem tissues, and hence are called open vascular bundles. In the monocotyledons, the vascular bundles have no cambium present in them. Hence, since they do not form secondary tissues, they are referred to as closed. When xylem and phloem within a vascular bundle are arranged in an alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundles, the xylem and phloem are jointly situated along the same radius of vascular bundles. Such vascular bundles are common in stems and leaves. The conjoint vascular bundles usually have the phloem located only on the outer side of xylem. Xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue organization of roots, stems and leaves, it is convenient to study the transverse sections of the mature zones of these organs. 6.3.1 Dicotyledonous root. Look at figure 6.6a. It shows the transverse section of the sunflower root. The internal tissue organization is as follows. The outermost layer is epibulma. Many of the cells of epibulma protrude in the form of unicellular root hairs. The cortex consists of several layers of thin-walled parenchyma cells. With intercellular spaces. The innermost layer of the cortex is called endodermis. It comprises a single layer of barrel-shaped cells without any intercellular spaces. The tangential as well as radial walls of the endodermal cells have a deposition of water-impermeable, waxy material suberin in the form of Casparian strips. Next to endodermis lies a few layers of thick-walled parenchymatous cells referred to as pericycle. Initiation of lateral roots and vascular cambium during the secondary growth takes place in these cells. The pith is small or inconspicuous. The parenchymatous cells which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissues on the inner side of the endodermis such as pericycle, vascular bundles and pith constitute the stellae. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. Figure 6.6b. It has epidermis, cortex, endodermis, pericycle, vascular bundles and pith. As compared to the dicot root which have fewer xylem bundles, there are usually more than 6 polyak xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous roots do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of a typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Figure 6.7a. Covered with a thin layer of cuticle, it may bear trichomes and a few stomata. The cells arranged in multiple layers between epidermis and pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of parenchymatous cells just below the epidermis, which provide mechanical strength to the young stem. Cortical layers below hypodermis consist of rounded thin-walled parenchymatous cells with conspicuous intercellular spaces. The innermost layer of the cortex is called the endodermis. The cells of the endodermis are rich in starch grains and the layer is also referred to as the starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. In between the vascular bundles there are a few layers of radially placed parenchymatous cells, which constitute medullary rays. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundles is a characteristic of dicot stem. Each vascular bundle is conjoint, open, and with endoc protoxylem. A large number of rounded, parenchymatous cells with large intercellular spaces which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by a sclerenchymatous bundle sheath, and a large, conspicuous parenchymatous ground tissue. Figure 6.7b. Vascular bundles are conjoint and closed. Peripheral vascular bundles are generally smaller than the centrally located ones. The phloem parenchyma is absent, and water-containing cavities are present within the vascular bundles. 6.3.5 Dorsiventral dicotyledonous leaf. The vertical section of a dorsiventral leaf through the lamina shows three main parts, namely, epidermis, mesophyll and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf, has a conspicuous cuticle. The abaxial epidermis generally bears more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and the lower epidermis is called the mesophyll. Mesophyll, which possesses chloroplasts and carry out photosynthesis, is made up of parenchyma. It has two types of cells, the palisade parenchyma and the spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells, which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cells and extends to the lower epidermis. There are numerous large spaces and air cavities between these cells. Vascular system includes vascular bundles, which can be seen in the veins and the midrib. The size of the vascular bundles are dependent on the size of the veins. The veins vary in thickness in the reticulate venation of the dicot leaves. The vascular bundles are surrounded by a layer of thick-walled bundle sheath cells. Look at figure 6.8a and find the position of xylem in the vascular bundle. 6.3.6 Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsiventral leaf in many ways. It shows the following characteristic differences. In N. On both the surfaces of the epidermis. And the mesophyll is not differentiated into palisade and spongy parenchyma. Figure 6.8b. In grasses, certain adaxial epidermal cells along the veins modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaves curl inwards to minimize water loss. The parallel venation in monocot leaves is reflected in the near similar sizes of vascular bundles except in main veins, as seen in vertical sections of the leaves. 6.4 Secondary growth. The growth of the roots and stems in length with the help of apical meristem is called the primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called the secondary growth. The tissues involved in secondary growth are the two lateral meristems, vascular cambium and cork cambium. 6.4.1 Vascular cambium. The meristematic layer that is responsible for cutting off vascular tissues, xylem and phloem, is called vascular cambium. In the young stem it is present in patches as a single layer between the xylem and phloem. Later it forms a complete ring. 6.4.1.1 Formation of cambial ring. In dicot stems, the cells of cambium present between primary xylem and primary phloem is the intrafascicular cambium. A cells of medullary rays, adjoining these intrafascicular cambium become meristematic and form the interfascicular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of the cambial ring. The cambial ring becomes active and begins to cut off new cells, both towards the inner and the outer sides. The cells cut off towards pith mature into secondary xylem, and the cells cut off towards periphery mature into secondary phloem. The cambium is generally more active on the inner side than on the outer. As a result, the amount of secondary xylem produced is more than secondary phloem and soon forms a compact mass. The primary and secondary phloems get gradually crushed due to the continued formation and accumulation of secondary xylem. The primary xylem, however, remains more or less intact, in or around the center. At some places, the cambium forms a narrow band of parenchyma, which passes through the secondary xylem and the secondary phloem in the radial directions. These are the secondary medullary rays, figure 6.9. (4.1.3) Spring wood and autumn wood. The activity of cambium is under the control of many physiological and environmental factors. In temperate regions, the climatic conditions are not uniform through the year. In the spring season, cambium is very active and produces a large number of xylary elements having vessels with wider cavities. The wood formed during this season is called spring wood or early wood. In winter, the cambium is less active and forms fewer xylary elements that have narrow vessels, and this wood is called autumn wood or late wood. The spring wood is lighter in color and has a lower density, whereas the autumn wood is darker and has a higher density. The two kinds of woods that appear as alternate concentric rings constitute an annual ring. Annual rings seen in a cut stem give an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compounds like tannins, resins, oils, gums, aromatic substances and essential oils in the central or innermost layers of the stem. These substances make it hard, durable and resistant to the attacks of microorganisms and insects. This region comprises dead elements with highly linified walls and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The peripheral region of the secondary xylem is lighter in color and is known as the sapwood. It is involved in the conduction of water and minerals from root to leaf. 6.4.2 Cork cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layers get broken and need to be replaced to provide new protective cell layers. Hence, sooner or later, another meristematic tissue called cork cambium or phellogen develops, usually in the cortex region. Phellogen is a couple of layers thick. It is made of narrow, thin-walled and nearly rectangular cells. Phellogen cuts off cells on both sides. The outer cells differentiate into cork or phellum while the inner cells differentiate into secondary cortex or phelloderm. The cork is impervious to water due to suberin deposition in the cell wall. The cells of secondary cortex are parenchymatous. Phellogen, phellum, and phelloderm are collectively known as pariderm. Due to activity of the cork cambium, pressure builds up on the remaining layers peripheral to phellogen, and ultimately these layers die and slough off. Bark is a non-technical term that refers to all tissues exterior to the vascular cambium, therefore including secondary phloem. Bark refers to a number of tissue types, viz. pariderm and secondary phloem. Bark that is formed early in the season is called early or soft bark. Towards the end of the season, late or hard bark is formed. Name the various kinds of cell layers which constitute the bark. At certain regions, the phellogen cuts off closely arranged parenchymatous cells on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped openings called lenticels. Lenticels permit the exchange of gases between the outer atmosphere and the internal tissue of the stem. These occur in most woody trees, figure 6.10. 6.4.3 Secondary growth in roots. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem bundles, a portion of pericycal tissue, above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular, figure 6.11. Further events are similar to those already described above for a dicotyledon stem.